Today, we're diving into the fascinating world of muscle tissue, exploring its common properties, functions, and intricate organization. This lecture draws extensively from the comprehensive material presented by Professor Vanessa. Muscle tissue is a primary tissue type, fundamentally responsible for contraction and movement. We have three main types of muscle in our bodies. Skeletal muscle moves the body by pulling on bones. Cardiac muscle, found exclusively in the heart, pumps blood through the cardiovascular system. Lastly, smooth muscle pushes fluids and solids through our internal passageways and organs.
All muscle tissues share several common properties. First, there's excitability, which is their ability to receive and respond to stimuli. Then, contractility, the unique ability of muscle cells to shorten. Extensibility allows muscles to stretch. And finally, elasticity is the ability of the muscle to recoil to its original resting length after stretching.
Skeletal muscle, specifically, serves a variety of crucial functions. It produces movement by pulling on tendons, which in turn move bones. It's essential for maintaining our posture and overall body position. Skeletal muscle also supports soft tissues and guards body entrances and exits, such as those of the urinary and digestive tracts. Another vital function is maintaining body temperature. Working muscles generate and release heat. Finally, skeletal muscles act as a significant storage site for nutrients, serving as a source of proteins and amino acids. Let's move on to the organization of skeletal muscle at the tissue level. Skeletal muscles, the muscles attached to our bones, are complex structures. They contain skeletal muscle tissue itself, along with connective tissues, blood vessels, and nerves. These muscles have three distinct layers of connective tissue. The epimysium is an outer layer of collagen fibers that surrounds the entire muscle and separates it from surrounding tissues, connecting to the deep fascia. Inside the epimysium, the perimysium surrounds individual bundles of muscle fibers called fascicles. This layer contains collagen and elastic fibers, blood vessels, and nerves. Finally, the endomysium surrounds each individual muscle cell or muscle fiber, loosely interconnecting them. This innermost layer contains capillary networks, nerve fibers, and special stem cells called myosatellite cells, which help repair damage. Importantly, the collagen fibers from the epimysium, perimysium, and endomysium converge at the ends of muscles. They form either tendons, which are bundles, or aponeuroses, which are broad sheets, both attaching skeletal muscles to bones. Skeletal muscles also boast extensive networks of blood vessels, which deliver oxygen and nutrients while removing metabolic wastes. A key characteristic is that skeletal muscles contract only when stimulated by the central nervous system. Branches of neuronal axons innervate individual muscle fibers. This is why they're often called voluntary muscles, though some, like the diaphragm, work subconsciously. Now let's explore the characteristics of skeletal muscle fibers and the components of a sarcomere. Skeletal muscle fibers, or muscle cells, are quite large and multinucleate. They develop from the fusion of embryonic cells called myoblasts. These cells are also known as striated muscle cells because of visible striations, which are due to the organized arrangement of myofibrils. The sarcolemma is the plasma membrane of a muscle fiber, enclosing the sarcoplasm, which is the cytoplasm. It maintains a negative resting membrane potential, and a change in this potential is what initiates a contraction. Transverse tubules, or T-tubules, are narrow tubes continuous with the sarcolemma that extend deep into the sarcoplasm. They're filled with extracellular fluid and quickly transmit electrical impulses, known as action potentials, from the sarcolemma into the cell interior. This ensures a coordinated contraction of the entire cell. Another critical component is the sarcoplasmic reticulum, or SR, a tubular network similar to the smooth endoplasmic reticulum. It surrounds each myofibril and forms chambers called terminal cisternae, which attach to the T-tubules. A triad consists of two terminal cisternae flanking a T-tubulum. The SR is specialized for the storage and release of calcium ions, which are actively transported from the cytosol into the terminal cisternae. Myofibrils are organized collections of contractile protein filaments responsible for muscle contraction. They run the entire length of the muscle fiber. Myofilaments themselves are bundles of contractile protein filaments. We have two types: thin filaments, primarily composed of actin, and thick filaments, primarily composed of myosin. Sarcomeres are the repeating structural and functional units of a myofibril. The thin and thick filaments within them are arranged in a very specific way, and their interactions are what produce contraction. Sarcomeres are the smallest contractile units of the muscle fiber. Each sarcomere contains thin filaments, thick filaments, and proteins that stabilize and regulate the interactions between these filaments. When we look at a sarcomere, we see distinct bands. The A bands are dark bands that span the length of the thick filaments. Within the A band, there's an M line, a vertical line in the center that stabilizes the thick filaments. The H band, found on either side of the M line, contains only thick filaments. The zone of overlap is where thick and thin filaments interdigitate. The I bands are light bands, containing only thin filaments. They extend from the A band of one sarcomere to the A band of the next sarcomere. Z lines bisect the I bands and mark the boundaries between adjacent sarcomeres. Titin, an elastic protein, extends from the tips of thick filaments to the Z line, helping to keep filaments aligned and restore resting sarcomere length. Delving into the composition of thin filaments, we find filamentous actin, or F-actin, the twisted strand of globular G-actin molecules. The active sites on these G-actin molecules are where myosin binds. Nebulin holds the F-actin strands together. Tropomyosin covers the active sites on G-actin, preventing actin-myosin interaction. Troponin is a globular protein that holds tropomyosin in place and binds to calcium ions. When calcium ions are present, troponin shifts tropomyosin off the actin active sites, allowing contraction to begin.
Thick filaments contain myosin. Each myosin molecule has a tail, which binds to other myosin molecules, with tails pointing toward the M line. The head, made of two globular protein subunits, projects toward the nearest thin filament. Each thick filament also has a titin core, acting as a molecular spring to recoil and return the sarcomere to its resting length.
This leads us to the sliding filament theory. During contraction, the thin filaments slide toward the center of the sarcomere, specifically the M line, alongside the thick filaments. As a result, the H band and I bands narrow, the zones of overlap widen, and the Z lines move closer together. Crucially, the width of the A band remains constant.
Now let's detail the events involved in the neural control of skeletal muscle contraction and relaxation. All cells maintain a negative resting membrane potential. For skeletal muscle cells, this is around negative 85 millivolts. Stimuli can change the local distribution of ions across the plasma membrane, altering the membrane potential. Depolarization occurs when the membrane potential becomes less negative due to an influx of sodium ions. Hyperpolarization makes it more negative due to potassium ion outflow. Repolarization is the return to the resting membrane potential after depolarization. Neurons and muscle cells have electrically excitable membranes and can produce action potentials that propagate along the plasma membrane. The neuromuscular junction, or NMJ, is the synapse between a motor neuron and a skeletal muscle fiber. Skeletal muscle contracts only when stimulated by motor neurons. The axon terminal, the expanded end of the motor neuron's axon, releases a neurotransmitter, a chemical, into the synaptic cleft, the narrow space between the axon terminal and the motor end plate. The motor end plate is the folded membrane of the skeletal muscle fiber at the NMJ. When an action potential arrives at the axon terminal, it triggers the release of acetylcholine, or ACH, the neurotransmitter at the NMJ. ACH then diffuses across the synaptic cleft and binds to chemically gated sodium-ion channels on the motor end plate. This binding opens the channels, causing sodium ions to rush into the cell and depolarize the motor end plate, generating an action potential. ACH is removed from the synaptic cleft either by diffusion or by being broken down by acetylcholine esterase, or ACHE, which closes the receptor channels.
Next comes excitation-contraction coupling, the link between the action potential in the sarcolemma and the start of muscle contraction. The action potential travels down the T-tubules to the triads, triggering the release of calcium ions from the SR. These calcium ions bind to troponin on the thin filaments. Troponin changes position, moving tropomyosin off the active sites on actin, allowing contraction to begin. The contraction cycle is a series of molecular events. It involves the formation of cross bridges, where myosin heads bind to the active sites on actin. Then, during the power stroke, the myosin head uses energy from ATP to pivot and pull the active sites toward the M line. The entire cycle repeats as long as calcium ion concentrations remain elevated and ATP reserves are sufficient. Once the stimulus is removed, calcium channels in the SR close, and calcium ion pumps return calcium to the SR. Troponin then shifts, covering the active sites and preventing further cross-bridge formation. When muscle cells contract, they produce tension, a pulling force. For movement to occur, this tension must overcome the load or resistance. At any given time, some myosin heads are performing a power stroke, while others are being reactivated. The entire muscle shortens uniformly because all sarcomeres contract together. The speed of shortening depends on the cycling rate, which is the number of power strokes per second.
Muscle relaxation depends on several factors: the duration of stimulation at the NMJ, the presence of free calcium ions in the cytosol, and the availability of ATP. When the stimulus ends, ACH is broken down by ACHE, and calcium ions return to the SR via active transport. As a result, calcium ions detach from troponin. Troponin returns to its original position, and the active sites on actin are covered by tropomyosin again. An interesting phenomenon related to muscle contraction is rigor mortis, the stiffening of muscles after death. This occurs because, without ATP, ion pumps for active transport cease to function. Calcium ions remain in the cytosol, triggering a sustained contraction. Since cross bridges cannot detach without ATP, the myosin heads remain attached to actin, locking the muscles in a contracted position. Let's explore the mechanisms responsible for the different amounts of tension produced in a muscle fiber. Tension is the pull force generated by a muscle when it contracts. The amount of tension an individual muscle fiber produces depends on three factors: the number of power strokes performed, the resting length of the muscle fiber at the time of stimulation, which we call the length-tension relationship, and the frequency of stimulation. The length-tension relationship states that the tension produced by a muscle fiber relates to the length of its sarcomeres. The number of power strokes performed by cross bridges depends on the amount of overlap between thick and thin filaments, or the zone of overlap. Maximum tension occurs when the maximum number of cross bridges form at the optimal sarcomere length. If there's no zone of overlap, no tension can be produced. Conversely, if a sarcomere is too short and cannot shorten further, no tension can be produced even with a large zone of overlap. The frequency of stimulation also plays a crucial role. A twitch is a single stimulus contraction relaxation sequence lasting typically between 7 and 100 milliseconds. Muscles in the body rarely receive just one stimulus, so twitches are mostly observed in laboratory settings. Muscular contractions generally require many repeated stimuli. A myogram is a graph that illustrates tension development in muscle fibers. A twitch has three phases: the latent period, where stimulation occurs but no tension is yet produced;
The contraction phase, where tension increases as cross bridges form and myosin heads perform power strokes, and the relaxation phase, where tension decreases as calcium ion levels fall, myosin binding sites on actin are covered, and cross bridges detach.
When repeated stimulations occur after the end of each relaxation phase, but the stimulus frequency is less than 50 per second, we see TRPE, an increase in peak tension. This produces a series of contractions with increasing tension and is typically seen in cardiac muscle, not skeletal muscle. Wave summation, however, occurs when successive stimuli arrive before the relaxation phase has completed, leading to increasing tension. This happens when the stimulus frequency is greater than 50 per second. Tetanus represents maximum tension. In incomplete tetanus, a muscle produces near maximum tension due to rapid cycles of contraction and very brief, incomplete relaxation. Complete tetanus is when the muscle is in continuous contraction due to a high stimulation frequency, completely eliminating the relaxation phase. The disease tetanus, caused by the neurotoxin of the bacterium Clostridium tetani, results in sustained, powerful muscle contractions and is often fatal. Next, let's compare the different types of skeletal muscle contraction. The tension produced by an entire skeletal muscle depends on the number of stimulated muscle fibers. A motor unit consists of a motor neuron and all the muscle fibers it controls. This can range from a few fibers to thousands, and all fibers in a motor unit contract simultaneously. Smaller motor units allow for finer control of movement. A fasciculation is an involuntary muscle twitch caused by the synchronous contraction of one motor unit. Recruitment is the increase in muscle tension that results from an increase in active motor units. This produces a smooth, steady increase in tension. Maximum tension is achieved when all motor units reach complete tetanus, though this can only be sustained for a very short time. Sustained contractions, which produce less than maximum tension, often rely on motor unit summation, a relay approach where some motor units contract while others rest and recover. Muscle tone refers to the resting tension of a skeletal muscle. Without causing movement, motor units actively stabilize the positions of bones and joints, maintaining balance and posture. Elevated muscle tone also increases resting energy consumption. We categorize muscle contractions into two main types. Isotonic contractions cause the skeletal muscle to change length. These can be concentric, where muscle tension exceeds the load and the muscle shortens. The speed of shortening depends on the load's size. Or they can be eccentric, where peak muscle tension is less than the load and the muscle elongates due to gravity or the contraction of another muscle. In contrast, isometric contractions mean the skeletal muscle develops tension but does not change length. The tension produced never exceeds the load. It's also important to remember that the load and speed of contraction are inversely related. The heavier the load, the slower the contraction, and the longer it takes for the muscle to shorten and begin movement. For each muscle, an optimal combination of tension and speed exists for any given load.
Muscle relaxation and the return to resting length are passive processes. This is due to elastic forces, where tendons recoil and help return muscle fibers to resting length. Opposing muscle contractions can also return a muscle to resting length quickly. Finally, gravity assists opposing muscles in returning muscles to their resting length.
Let's investigate how muscle fibers obtain the energy to power contractions. Normal muscle function requires substantial intracellular energy reserves, a normal circulatory supply, a normal blood oxygen level, and a normal blood pH. Any interference with these factors reduces the efficiency of muscle contraction and can lead to premature fatigue. ATP, or adenosine triphosphate, is the only energy source directly used for muscle contraction. Contracting muscles use a significant amount of ATP. While muscles do store some ATP, these stores are depleted within 4 to 6 seconds in active muscles. Therefore, more ATP must be generated to sustain a contraction. At rest, skeletal muscle fibers produce more ATP than they need. This excess ATP transfers energy to creatine to create creatine phosphate, or CP. The energy stored in CP is then used to convert ADP back to ATP during a contraction, a process catalyzed by the enzyme creatine kinase. Energy stored as CP is typically sufficient for about 15 seconds of sustained contraction.
The other main way ATP is generated is through aerobic metabolism, an oxygen-dependent process where mitochondria use organic substrates like pyruvate to produce ATP. This is the primary ATP source for resting muscles and includes the citric acid cycle and electron transport chain. It's very efficient, generating 15 ATP molecules for every pyruvate molecule. Resting muscles break down fatty acids from circulation as an energy source, while active muscles break down pyruvate from glycolysis. Muscle metabolism adapts to varying levels of activity. At rest, energy requirements are low and oxygen availability is high, so muscles metabolize fatty acids and store glycogen and CP. During moderate activity, ATP demand increases, but oxygen is still available, so muscles primarily generate ATP through the aerobic breakdown of glucose. However, at peak activity, the demand for ATP is very high, and oxygen supply often cannot keep up. In this scenario, muscles generate about 1/3 of their ATP via aerobic metabolism, with the rest coming from glycolysis. Pyruvate that doesn't enter the mitochondria converts to lactic acid. The buildup of hydrogen ions and lactic acid from anaerobic metabolism can then cause muscle fatigue. The recovery period is the time required for muscles to return to pre-exertion conditions. During exertion, lactate is transferred from muscles to the liver, where it's converted to pyruvate. 70 to 80% of these pyruvate molecules are then converted back to glucose, which returns to the muscle to help rebuild its glycogen reserves. This is known as the Cori cycle.
Oxygen debt, or excess post-exercise oxygen consumption, EPOC, is the amount of oxygen needed to restore normal pre-exertion conditions. After exercise, the body needs more oxygen than usual, leading to increased breathing rate and depth. Better fitness can reduce the duration of EPOC. Active skeletal muscles also produce heat, releasing up to 85% of the heat needed to maintain normal body temperature. If too much heat is generated, as during peak activity, body temperature climbs and heat loss at the skin accelerates. Hormones also influence muscle metabolism. Growth hormone and testosterone stimulate protein synthesis and skeletal muscle enlargement. Thyroid hormones elevate the rate of energy consumption in both resting and active muscles. Epinephrine stimulates muscle metabolism, increasing both the duration of stimulation and the force of contraction.
Let's relate the types of muscle fibers to muscle performance and discuss muscle hypertrophy, atrophy, and aging, along with how physical conditioning affects muscle tissue. Muscle performance is characterized by force, the maximum tension produced, and endurance, the amount of time an activity can be sustained. Both force and endurance depend on the type of muscle fibers and physical conditioning. Skeletal muscle fibers come in different types. Fast fibers, which make up the majority of skeletal muscle fibers, contract quickly. They are large in diameter, have many myofibrils and large glycogen reserves, but few mitochondria. They produce strong contractions with high force, but fatigue quickly, giving them low endurance. Muscles dominated by fast fibers are often called white muscles. Slow fibers, on the other hand, contract more slowly. They are small in diameter, have numerous mitochondria, a high oxygen supply from an extensive capillary network, and contain myoglobin, a red pigment that binds oxygen. They produce less force, but have much greater endurance. Muscles dominated by slow fibers are called red muscles. Intermediate fibers are mid-sized and are slower to fatigue than fast fibers, though they contain little myoglobin.
Most human muscles contain a mixture of these fiber types. Muscle hypertrophy is the growth of muscle from heavy training. This involves an increase in the diameter of muscle fibers, the number of myofibrils, the number of mitochondria, and glycogen reserves. Crucially, the number of muscle fibers does not significantly change, but they grow in size. Conversely, muscle atrophy is a reduction of muscle size, tone, and power due to lack of activity. If moderate, it's reversible. However, if severe and muscle fibers die, they are not replaced.
As we age, muscle tissue undergoes changes. Skeletal muscle fibers become smaller in diameter, and skeletal muscles become less elastic, a process called fibrosis, which is an increase in fibrous connective tissue. Tolerance for exercise decreases, and the ability to recover from muscular injuries diminishes, largely due to a decrease in the number of satellite stem cells. Muscle fatigue occurs when muscles can no longer perform at a required level. This correlates with depletion of metabolic reserves, damage to the sarcolemma and sarcoplasmic reticulum, a decline in pH that affects calcium ion binding and alters enzyme activities, and weariness due to low blood pH and pain. Physical conditioning significantly improves both power and endurance. Anaerobic endurance refers to the length of time muscle contraction can be supported by stored ATP, CP, and ATP produced by glycolysis. Conditioning improves muscle power, though endurance is limited by available ATP, CP, and glycogen, as well as the ability to tolerate lactate buildup and pH decrease. Fatigue typically sets in within 2 minutes of maximal activity. This type of conditioning involves fast fibers and stimulates hypertrophy, and it's improved by frequent, brief, intensive workouts.
Aerobic endurance is the length of time muscle contraction can be supported by ATP produced by mitochondria. It doesn't stimulate muscle hypertrophy, but requires a good blood supply to deliver nutrients and oxygen. Training for aerobic endurance involves sustained low levels of activity, allowing for prolonged activity. Cross-training combines both aerobic and anaerobic exercises. Improvements in aerobic endurance result from alterations in the characteristics of muscle fibers, often an increase in the proportion of slow fibers, and improvements in cardiovascular performance, leading to better blood flow and an increased number of capillaries.
Finally, let's identify the structural and functional differences between skeletal muscle fibers, cardiac muscle cells, and smooth muscle cells. Cardiac muscle tissue is found exclusively in the heart. Cardiac muscle cells are small, branched, striated, and typically have a single nucleus. They have excitable membranes, short, wide T-tubules, but no triads or terminal cisternae in their SR. They are almost totally dependent on aerobic metabolism, containing lots of myoglobin and many mitochondria. They contact each other via intercalated discs. Intercalated discs are specialized connections that join the sarcolemmas of adjacent cardiac muscle cells through gap junctions and desmosomes. Their functions include stabilizing the position of adjacent cells, maintaining the three-dimensional structure of cardiac muscle tissue, and allowing ions to move from one cell to another, which ensures cardiac muscle cells beat in rhythm. Functionally, cardiac muscle exhibits automaticity, meaning it can contract without neural stimulation. Pacemaker cells generate the electrical signals that initiate contraction, though the nervous system can alter the pace and tension. Cardiac contractions are longer than those in skeletal muscle, have longer refractory periods, and are resistant to fatigue. They require both intracellular and extracellular calcium ions, and critically, cardiac muscle does not experience wave summation or tetanic contractions.
Smooth muscle tissue is found around other tissues and internal organs throughout the body. In the integumentary system, erector pili muscles erect hairs. In the cardiovascular and respiratory systems, smooth muscle around blood vessels and airways regulates blood pressure and airflow.
In the digestive and urinary systems, smooth muscle moves materials along and out of the body and forms sphincters. In the reproductive system, smooth muscle help transport gametes and are responsible for the delivery of a fetus. Structurally, smooth muscle cells are long, slender, spindle-shaped cells with a single central nucleus. They have no T-tubules, myofibrils, or sarcomeres, making them non-striated muscle. They have scattered thick filaments with many myosin heads, and thin filaments are attached to dense bodies. These dense bodies connect adjacent cells, transmitting contractions throughout the tissue. Smooth muscle also lacks tendons or aponeuroses. Functionally, smooth muscle differs from other muscle tissue in its excitation-contraction coupling, length-tension relationships, control of contractions, and smooth muscle tone. In excitation-contraction coupling, entry of calcium ions from extracellular fluid and their release from the SR trigger contraction. These calcium ions bind the protein calmodulin, which then activates the enzyme myosin light chain kinase, allowing myosin heads to attach to actin. Due to the lack of sarcomeres, tension and resting length are not directly related in smooth muscle. Even a stretched smooth muscle can contract, a property known as plasticity, its ability to function over a wide range of lengths.
Regarding control of contractions, multi-unit smooth muscle cells are innervated in motor units, and each cell may connect to more than one motor neuron. Visceral smooth muscle cells, however, are not connected to motor neurons. They are arranged in sheets or layers, and their rhythmic cycles of activity are controlled by pace-setter cells. Smooth muscle tone refers to a normal background level of activity, which can be decreased by neural, hormonal, or chemical factors.
From the intricate organization of skeletal muscle fibers to the unique properties of cardiac and smooth muscle, these tissues are fundamental to nearly every bodily function, constantly adapting to our needs and environment. If you found this discussion illuminating, please share it with others.